All right, so welcome back. So this is going to be part three in our cells lecture. And in this part, we're going to look at the plasma membrane in a little bit more detail. So this plasma membrane is a biological membrane. And the significance of the membrane is that it's going to help keep things that are on the inside of the cell in, and it's going to help keep things on the outside of the cell out. Specifically, prevent them from just freely flowing back and forth. We will see that things on the outside can come in and things that are on the inside can go out, but they have to do so in a particular way, okay, or particular ways, depending on the nature of what's moving. So, to start off, we said earlier that our biological membrane, in the sense of our cells, is known as a plasma membrane, and we called it a phospholipid bilayer, right? For this reason, down here, you have two layers opposed against each other, two layers of phospholipids, right? We said that phospholipids had a polar head and a non-polar tail. The head was hydrophilic, liked to be in contact with water, and non-polar tails did not like to be in contact with water. Okay. Now, I should note the phospholipids, these are not living things. Okay. These are biomolecules. Okay. These are not living. The head, which is hydrophilic, does not really want, quote unquote, want to be in contact with water. Likewise, the non-polar tail doesn't not like to be in contact with water. It doesn't fear water. Okay. That is that's just our way of saying it's in contact with water or it's not in contact with water. It quote unquote wants to be or quote unquote does not want to be. Okay. So we're trying not to be anthropomorphic here. Okay. So we're not trying to ascribe human qualities onto non-human things, right? Phospholipids are not human. They're not living. So they don't want to do anything or they don't not want to do something. That's just what we're calling it. Okay, so keep that in mind as we're moving forward. All right. Now, this membrane, this phospholipid bilayer, there's another term that is used to describe it, and this term is semi-permeable. Sometimes you'll see the term selectively permeable. Okay. Now, I personally like the term selectively permeable more than semi-permeable. Right. The reason for this is semi-permeable. The term semi-permeable, when describing the membrane, gives the kind of impression that it's kind of permeable, but it's not fully, so it's only semi-permeable. Okay. The term selectively permeable gives the impression that it is fully permeable, but only to selective things, or it's selective in what it lets across and how it lets that thing across. Okay, so I prefer the use of the term selectively permeable when describing the nature of the plasma membrane. Okay. Now, the way that it lets things across, things can diffuse freely across it. So just go directly from one side to the other and go straight through the membrane. Sometimes they have to go through protein channels or protein carriers to help to get from one side to the next. Okay, so the selective permeability regulates what gets in the inside and what leaves and goes out to the outside. So when we look here, we can see what things can freely go through this plasma membrane and what things cannot freely go through. Now, these things can still go through, but they have to go through a channel. All right. So the first class are permeable molecules, or things that are permeable to the membrane. They can go freely through it. These are small and non-polar. Okay, small and non-polar molecules. Okay, so things like gases, carbon dioxide, oxygen. Things that are steroid based, right? So we said in the last part, in part two, that the plasma membrane is made of phospholipids, right? Lipids are lipids, right? So things that are also made from lipids, like steroid based hormones, testosterone, estrogen, things like that, can freely diffuse through it because of this idea that like dissolves like. Similar things dissolve in things that are similar to it. Okay. If it is impermeable, it means that it cannot freely go through the cell. Okay. That doesn't mean that it can't get to the other side of the cell, can't get to the inside or to the outside, but it can't do so freely. Okay. So it has to do so through the use of a protein. And these are either going to be what are called carrier proteins or protein channels or something like that. Okay. So these are larger things: carbohydrates, proteins, charged ions like calcium, sodium, potassium, all of those other molecules that you may find in the blood, like or surrounding cells, like glucose and things like that, amino acids. All right, those cannot go freely through the membrane, so they have to go in or out through a channel. So when looking here, what's a feature of all of these cells, right? So all of these cells have a membrane in common, right? And this is one of the features of cells that we talked about in part one. They're all going to have a membrane, all right? So this membrane, specifically in our cells, is going to be what's called a fluid mosaic membrane. And this is referred to as the fluid mosaic model of the plasma membrane, okay? Meaning that it is fluid, so it's not rigid, it's not static, it's not just stiff like a board, okay? And then mosaic, meaning that it's not uniform in what it's made of, okay? It's not made of just one thing, but it's a mosaic of different molecules. So you have phospholipids, you have things that have carbohydrate groups attached, so glycoproteins, you have cholesterol, you have proteins that are on the inside of the membrane, you have proteins that are on the outside, you have proteins that span the whole width of the membrane, right? And so this fluid mosaic model is a very good description of this plasma membrane. If you look at this top kind of animation, you see that it kind of moves and kind of ripples like a wave. Okay, so you can see it moving there, and you can see that there's other things embedded within the membrane, right? We can also take the phospholipids. So the phospholipids in this example down here are all of these kind of red and orange structures with the head and the two fatty acid tails. We can take the phospholipids and modify them, and by modifying them, you change the fluidity of the membrane, or you change how much or to what degree it can move, all right? So the ways that we can alter this fluidity, we can alter a couple different things. One, we can alter the temperature, all right? As we increase the temperature in our environment, in the cellular environment, we can increase the fluidity, all right? So higher temperature equals fluidity increasing. Lower temperatures cause the membrane to become less fluid, all right? The reason for this is that an increasing temperature causes the phospholipids to move back and forth, and this causes them to separate and resulting in a little bit more fluid membrane, right? When they're colder, or when it's a colder environment, they pack closer together and you get less movement, okay? However, we can become too rigid, right? If we drop too low in the cell membrane, or the plasma membrane becomes too fixed and too stiff, okay? All right, we can also change the nature of the phospholipids themselves, right? So we said in the previous part, or part one, that the phospholipids can either be saturated or unsaturated. Now, this is describing the fatty acids that are part of the phospholipid, so part of the tails of the phospholipid molecules. When you look at them, if the tail is more or less straight, then that means that is a saturated fatty acid. And the saturation means, so a fatty acid is this carbon backbone here, and on either side, every carbon is bound to a hydrogen, okay, on all sides, okay. For an unsaturated, you notice that there are some carbons that only are bound to one hydrogen, whereas all of these others are bound to at least two, okay. So this right here represents an unsaturated fatty acid because it is not completely saturated with hydrogens, so you call it unsaturated. Okay, so what you get is you get a double covalent bond, so you get more sharing of electrons here, and that causes the shape, the three-dimensional shape of the fatty acid to change, and it changes from this linear shape in saturated fatty acids to this kind of kinked or crooked or bent shape in an unsaturated fatty acid, all right? Now, you can kind of use this to deduce the influence of the saturation on the membrane fluidity. If you have straight fatty acids, these can, let me go back, you can pack them together very, very quickly or very tightly, and the fluidity of the membrane is going to go down, right? It's going to decrease because you pack them very, very close together. If you have this bond and this kink in them, that causes the fatty acids and the phospholipids to not be quite as tightly adhered to each other, right? So they're not as closely packed, and so that causes the fluidity to increase a little bit more. Okay. You can get saturated fats from animal sources largely, and at room temperature they make it solid, so things like butter, right? And unsaturated you can get from plant sources, and at room temperature they do not form a solid, but they generally form a liquid. Okay. All right. We can also change the amount of cholesterol that we find in our plasma membrane. Okay. So cholesterol is a lipid derived, it's a lipid based molecule. All right. And it can pack tightly in between adjacent phospholipids, right? So you can deduce now that if we're packing the membrane with this cholesterol, the more cholesterol we pack in there, the tighter the membrane is going to be, the tighter those phospholipids are going to be opposed to each other. And so the less fluid it's going to be. So more cholesterol, the less fluidity you have in the membrane. Okay. Now, an important thing about cholesterol. Cholesterol is only from animal sources. Okay. Cholesterol does not come from plants. All right. So if you're at a grocery store, and you can look at this, you can test this for yourself the next time you go to the grocery store. Look and see what products are marked as cholesterol free, right? It's going to be a marketing strategy. They'll be marked as cholesterol free in an attempt to make you think, oh, this is cholesterol free, it's healthy, right? Because typically we think of high cholesterol, things like that, as being bad for you. Well, if whatever you're buying, whatever the food product is, doesn't have any animal products in it, of course it's going to be cholesterol free, because cholesterol only comes from animals, right? And if you don't have animals, you can't have cholesterol. Okay, so it's essentially a marketing strategy to make you think that by nature of not having cholesterol, it's automatically healthy. Okay, all right. And you can see here that with the changing in the nature of the phospholipids, saturated versus unsaturated, or the amount of cholesterol, we can change the rigidity and change the fluidity of the membrane here. Okay. All right. So for our different components of our cells here, and this is kind of leading into our next part, we have a plasma membrane. So what we just talked about, how we can influence its rigidity and fluidity. We have a nucleus on the inside, and then we have cytoplasm on the outside of the nucleus. So the membrane, we said plasma membrane, phospholipid bilayer made of phospholipids, but with other things as well. Nucleus on the inside with DNA and our genetic information, and then the cytoplasm on the outside. So cytoplasm is really cytosol. So cytosol is the fluid in the cell, the liquid part of the cell, plus all the organelles. So the ribosomes and the endoplasmic reticulum and the Golgi apparatus and the peroxisomes and all these other things. That's cytoplasm. So if you're referring to the cytosol, that's just the liquid. If you're referring to the cytoplasm, that's the liquid plus the organelles. So plus all those other things. All right. So what we're going to focus on now is we're going to move into looking specifically at the nucleus, and we're going to see how this can influence our develop of RNA from DNA, and then we'll use that RNA to make proteins in this process of protein synthesis, and that will be the topic of our next part.